Hello students, I welcome you all to E-Pathshala. This is Dr. P. A. Rajeshwari, Assistant Professor in the Department of Food Science and Nutrition, Avinashalingam Institute for Home Science and Higher Education for Women, Coimbatore. Today we are going to discuss about the module Food Starch. Starch is a plant polysaccharide, a major constituent of cereals, roots and tubers. Starch is found as discrete particles called granules as stored energy for growth and reproduction in plants. Other plant polysaccharides include cellulose, hemicellulose and pectins. Polysaccharides, also called as glycans, consist of monosaccharides bound to each other by glycosidic linkages. On hydrolysis, they yield the component monosaccharides. Polysaccharides that consist of one type of sugar structural units are called homoglycans, and those which contain several types of sugar units are called heteroglycans. The linkages of monosaccharides may be linear as in cellulose and amylose, or branched as in amylopectin and glycogen. The monosaccharide residue sequence may be periodic, one period containing one or several alternating structural units as in cellulose or hyaluronic acid. The sequence may contain shorter or longer segments with periodical arranged residues separated by non-periodic segments as in alginate and pectin, or the sequence may be non-periodic or long chain as in case of carbohydrate compounds in glycoproteins. Polysaccharides are widely distributed in nature and perform various roles such as structure, forming skeletal substances, example cellulose, hemicellulose and pectin in plants, and chitin mucopolysaccharides in animals. Assimilative reserve substance, example starch, dextrin, inulin in plants and glycogen in animals. Water binding substances such as agar, pectin and alginate in plants and mucopolysaccharides in animals. Now let us see about the various sources of starch. Cereals, legumes and other seeds are the primary sources of starch. Rice, wheat, maize and barley are the main providers of starch. Roots like arrowroot and tapioca and tubers like potato also yield starch to some extent. Let us move on to the starch and its appearance. Starch granules develop in cells called plastids during growth and maturation of the plant. They are synthesized by enzymes which unite simpler carbohydrate substrates forming long chain polymers. The organization of starch within granules as viewed under a microscope shows internal concentric shells. Starch granules vary in appearance, that is size and shape depending upon the source of starch. The size of starch granules ranges from 1 micron to 150 microns in diameter. Starch granules of rice are small and polygonal, while that of corn are a mixture of round and polygonal granules with overlapping sizes. The starch granules of tapioca are irregular, round, concave. The wheat granules are large, identified by oyster cell shape and appearance of distinct concentric rings or striations. Coming to the component of starch, the main component of starch is the amylase and amylopectin. Starch consists of a linear chain of glucose units called amylose as well as a branched chain of glucose units called amylopectin. If the amylase content of starch is less than 2%, it is called as waxy starch. Now let us see about the structure of amylose in detail. Amylose, it is a linear polymer of glucose units linked by alpha linkage. That is, the first carbon atom of the glucose connected to the hydroxyl group on the fourth carbon atom of the adjacent glucose unit. Amylase has a molecular weight of 500 to 5000 glucose units. It forms a helical structure and is responsible for the three-dimensional cell structures. It gives blue color with iodine solution, forming a single helix forming around the iodine molecule. It alters the cooking property and structure of starch as in thickened sauces and is dispersible in hot water. Next we will see about amylopectin. It is the branched polymer and which accounts for 72 to 80% of the total starch. It is composed of glucose subunits linked by alpha 1:4 and alpha 1:6 linkages. It has a molecular weight of 5000 to 50,000 glucose units. Crystallinity of raw starch granules is due to the alignment of linear chain segments of amylopectin. It gives red color with iodine. It is dispersible in water but forms a thick paste which does not set to a gel. Well students, now we will have a brief explanation about the physiochemical properties of starch. Most native starch granules show no obvious changes in water at temperatures below 60 degrees Celsius. As the temperature is raised by heating, the starch granules take up water and swell to many times of their original size. Amylose molecules are freed and move into the surrounding liquid medium. Next comes gelatinization. Gelatinization is an integral part of the heat treatment of starchy foods. It includes hydration, melting and swelling of the starch granules. Separate events but occur almost simultaneously during heating of starch in excess of temperatures of about 60 degrees Celsius to 70 degrees Celsius, and increase in clarity and increase in viscosity is obtained. Starch concentration of 3 to 10% is usually used. The process of gelatinization can be explained as follows. Polysaccharides have large numbers of hydroxyl groups that attract water. When the hydrated polysaccharide moves about in solution, bombarded by molecules of solute, it seeps through a large spherical volume. Thus, the hydrated molecule, even in lower concentration, comes in contact with another and gives the solution, as is the viscosity. As concentration of the polysaccharide increases, viscosity increases. The swollen granule, now chiefly amylopectins, is unfavorable for viscosity. When amylase, the hydrated linear polysaccharide molecules, twist and turn in solution, they collide with each other and shear the water layer and adhere to one another and form bonds relating in an interwoven meshwork that holds water both through attraction of hydroxyl groups and not involved in bonding, and also by means of the porous structure of three-dimensional network similar to gel. During cooling, at the completion of gelatinization, if stirring is continued, the solution becomes more viscous if it is not allowed to stand without stirring. Intermolecular bonds form resulting in the meshwork. If the starch is one of the waxy variety containing only branched amylopectin molecules, the intermolecular association is too little to be of any permanence unless the paste is very concentrated, that is less than 30%. In amylase-rich starches, such intermolecular bonding occurs readily even at relatively lower concentrations. Let us now have a look at the characteristics of some food starches. The characteristics of some food starches are tabulated in the following table. The first column denotes the sources of starch, next followed by gelatinization temperature, and the next column explains the gel characteristics. Rice, and the temperature is 68 degrees Celsius to 78 degrees Celsius, and it gives a tender and opaque gel. When wheat is cooked at a temperature of 59 degrees Celsius to 64 degrees Celsius, it gives a fairly high elastic firm, highly elastic gel. When maize or corn is gelatinized at 62 degrees Celsius to 70 degrees Celsius, they give a stiff opaque gel which undergoes extensive retrogradation, especially when frozen and thawed. Potatoes when gelatinized at 58 degrees Celsius to 66 degrees Celsius, they give high viscosity. They tend to form high viscosity and low tendency to form gel, which gives a ropey paste. Tapioca when gelatinized at 52 degrees Celsius to 64 degrees Celsius give a viscous fluid usually modified or formed into partially gelatinized pearls or small pellets called sago. Ragi when gelatinized at 60 degrees Celsius to 70 degrees Celsius gives a stiff opaque gel. Varagu when gelatinized at 80 degrees Celsius to 88 degrees Celsius forms a low tendency to form a gel. Samai when gelatinized at 81 degrees Celsius to 83 degrees Celsius have low tendency to form a gel. Cholam when gelatinized at 80 degrees Celsius to 82 degrees Celsius gives a stiff opaque gel. Thinai when gelatinized at 78 degrees Celsius to 85 degrees Celsius gives a stiff opaque gel. Kambu when gelatinized at 76 degrees Celsius to 81 degrees Celsius gives a stiff opaque gel. Sago when gelatinized at 83 degrees Celsius to 90 degrees Celsius is highly viscous and forms a gel. Now we will have a look at the various factors affecting gelatinization and strength of starch gel. Temperature of gelatinization. The temperature range required to start gelatinization is 60 degrees Celsius to 70 degrees Celsius, and to complete gelatinization is around 90 degrees Celsius. However, continued heating results in more free amylose. Agitation at this stage may break granules and produce a stringly texture. Application of this phenomena is vivid in case of making ragi kali or ragi balls. Next is the effects of other ingredients on gelatinization. Sugar increases transparency and tenderness of starch gels. Raw milk gives a stiffer gel than pasteurized milk. Acidity. Addition of strong hydrochloric acid to starch gel causes hydrolysis and liquefies the gel. Citric acid, being weak, takes a long time for hydrolysis. Enzymes. Amylase added at 90 degrees Celsius to starch gel break down starch rapidly. The action may be useful in bread making. The starch in egg yolk may cause a weak gel if it is not denatured by boiling and the gel completely. Dry heat. Dry heating of starch gel causes browning of starch. That process is called as dextrinization and leads to loss of gel strength. Size of starch molecules. Larger granules like potato starch will gelatinize at low temperature around 55 degrees Celsius when compared to smaller granules like corn that require higher temperature. Sources of starch. The stiffness of gel will vary depending upon the source of starch and thus the amylase content. The order of decreasing stiffness of starch gel is corn, wheat, rice, potato, arrowroot and tapioca. Fat content. Defatted starch gives a less opaque paste with increased gel strength. Fat is used as the ingredient for tenderizing the gel. Hydrolysis. The enzymes that break down or hydrolyze starch into the constituent sugar are known as amylases. Alpha amylases are found in plants and in animals. Human saliva is rich in amylase and the pancreas also secretes the enzyme amylase. Individuals from populations with a high starch diet tend to have more amylase genes than those with low starch diets. Beta amylase cuts starches into maltose units. This process is important in the digestion of starch and is also used in brewing, where amylase from the skin of seed grains is responsible for converting starch to maltose. Dextrinization. If starch is subjected to dry heat, it breaks down to form dextrins called as pyrodextrins in this context. This breakdown process is known as dextrinization. Pyrodextrins are mainly yellow to brown in color and dextrinization is partially responsible for the browning of toasted bread. Now let us move on to the use of starch and starch products in cookery. The starch of cereals such as rice, wheat, ragi, bajra, maize and barley are the staple food items in our daily diet. Boiled whole rice is used as the main item of the South Indian menu, while wheat products, mainly wheat flour, are the chief items in the North Indian menus in the form of chapatis, puris, paratha, roti, naan, etc. In these wheat-based items, starch is the basic structural component as well as the flavoring agent, accounting for dextrinization of starch. Other wheat products include biscuits, cookies, cakes, puffs and pastries. Wheat products such as structural broken wheat, semolina, wheat flour and pasta products including vermicelli, noodles, macaroni and spaghetti are extensively used as breakfast items. Rice and ragi flour are used in the preparation of gruel, sticks and sevai, which come under breakfast items. Coming to modified and derived starches. Natural starches may be processed to change their functional characteristics either to enhance a desired characteristic or to eliminate a problem. Such modified starches play an important role in various food industries. Modified starches are defined as starches with one or more original characteristics altered by processing treatment to suit their application in food industry. Modification of starch can be affected by physical changes, degradation of starch molecule and the addition of a chemical group. The modified starches are widely used as thickening and binding agents. Properties of modified starches. Modified starches are stable carbohydrate polymers. They are white or off-white, tasteless and odorless, flakes, amorphous or coarse particles. They are soluble in cold water, alcohol, ether and chloroform. On heating in water, the granules usually begin to swell at 45 to 80 degrees Celsius and gelatinize completely at higher temperature. Pre-gelatinized starches hydrate in cold water. The functional properties of starches can be improved by modification methods such as cross-linking, stabilization, esterification, depolymerization and pre-gelatinization. Improvements in specific properties include increased dispersibility, enhancement of gel strength, reduction of gel synersis, improvement of stability to acid, heat and shear. Pre-gelatinized starches. Pre-gelatinized starches are cooked and dried starches which upon addition of cold water swell and thicken the mixture. Now let us have a look at the types of starch modification. The first column provides the details on types of modification, followed by the purpose of modification, and last one denotes the treatment. They are produced by drying starch suspensions or starch paste on heated rolls or by extrusion of semi-dried starches. They find application in convenient foods like instant pudding and dry blends like cake mixes, idli mixes, dokla mixes and so on. Coming to another important aspect, that is low viscosity starches or otherwise called as converted starches. Native starches cannot be used to produce paste at concentrations above 10%. This drawback has evolved the production of low viscosity starches by chemical treatment that results in the formation of some of the glycolic bonds. Such treatments include acid and heat treatment of dry starch, that is pyrodextrins, acid treatment of starch in water slurries, example acid modified starches, thin boiling starches and fluidity starches. Oxidizing agents includes oxidized starch. Enzyme treatment in bacterial alpha amylase. The acid hydrolyzed starches can be cooked to high viscous concentrations which retrograde and set to gel on cooling. Therefore, fluidity starches are used as gelling agents in the manufacture of gum jellies such as jelly beans, gummy beans and orange slices. Thin boiling high amylase gels are used in confectionery. Oxidized starches are produced by hydrolysis and oxidation of starch suspensions on treatment with oxidizing agents, usually hydrochlorate, at below gelatinization temperatures. Next we will see about cross-linked starches. Cross-linked starches result from the action of functional reagents causing increased viscosity and stability. The reagents react with more than one hydroxyl groups, thus linking one starch molecule with another. Cross-linking improves the mouthfeel of waxy starches. The cohesive, rubbery and elastic starch after cross-linking becomes smooth and creamy. Cross-linked starches are used as thickeners in custard, pie fillings, cream toppings and canned foods. Next we will see about stabilized starches. Stabilized starches are prepared by esterification or etherification of starch, which introduces new side chains onto the amylase molecule. This inhibits retrogradation and imparts increased viscosity and stability. They are desirable for fruit fillings and are also used as thickeners and stabilizers in gravies, bakery products, soup powders, sauces and puddings, etc. The application of modified starches by the food industry is not due to their source of nutrition but for the functional properties that they contribute. Some of the desirable traits that the modified starches contribute to processing industry are texture, mouthfeel, thickening, gelling, binding and stability. Well students, in this module we have learnt about these food starches and various types of food starches and their application in food industry. Starch is the most common carbohydrate in the human diet and is contained in many staple foods. The major sources of starch intake worldwide are the cereals, the root vegetables. As an additive for food processing, food starches are typically used as thickeners, extenders, emulsifiers, stabilizers and are exceptional binders in processed meat and stabilizers in foods such as puddings, custards, soups, sauces, gravies, pie fillings and salad dressings, and even also used in the manufacturing of noodles and pastas. Starch is widely consumed by humans as an inexpensive and stable available carbohydrate source, and much work has been performed on the structure, functionality and applicability of starches. Although conventional sources of starch such as corn and potato and other sources with improved properties are becoming significant to allow simpler and more robust processes. The feasibility of the use of new starches, that is from legume, seed and palm as renewable materials for commercial application may provide cost reduction of raw material in food industries. Little information is available on the structure, properties and practical applicability of non-conventional sources of starches. Thus, it is necessary that these starches should be studied to obtain and report their structural parameters, and information is required to gain competitiveness in an international scale of industries. Starch is an important ingredient for the food industry, and the researchers are making progress in discovering new details about its structure, functionality and its impact on our health. Nowadays, starch usage has become more common because of the wide use of it in food preservation and processing industry, because of the new formulations of ready to serve, ready to eat and ready to cook foods. Well, starch with reference to the above mentioned aspect has to be explored more to improve the nutritional and sensory quality of food. Thank you.